right, so welcome back. We're moving into chapter 32. This is part two. It won't be nearly as long as the part one. In part two, we're just going to go over key characteristics to help us understand evolutionary history or relatedness. So this is kind of an overview just to, again, help to explain why animals are separated based on some characteristics that we've already explored in part one. So, move forward. Okay, so key item to remember here, these are, again, we're just going to give a brief overview of key evolutionary history or relatedness characteristics. So then we can dig into our different animal phyla, kind of tackling them one phyla at a time in our future studies together. Just kind of a little bit of a break and a reminder of why do we study these things, and a reminder of why biodiversity is so important, and having somewhat of an understanding but a deeper appreciation for different organisms is really important and key. So hopefully this is a nice reminder of that.
Our planet's diverse, thriving ecosystems may seem like permanent fixtures, but they're actually vulnerable to collapse. Jungles can become deserts, and reefs can become lifeless rocks, even without cataclysmic events like volcanoes and asteroids. What makes one ecosystem strong and another weak in the face of change? The answer, to a large extent, is biodiversity. Biodiversity is built out of three intertwined features: ecosystem diversity, species diversity, and genetic diversity. The more intertwining there is between these features, the denser and more resilient the weave becomes. Take the Amazon rainforest, one of the most biodiverse regions on Earth due to its complex ecosystems, huge mix of species, and the genetic variety within those species. Here are tangled liana vines, which crawl up from the forest floor to the canopy, intertwining with treetops and growing thick wooded stems that support these towering trees. Helped along by the vines, trees provide the seeds, fruits, and leaves to herbivores such as the tapir and the agouti, which disperse their seeds throughout the forest so they can grow. Leftovers are consumed by the millions of insects that decompose and recycle nutrients to create rich soil. The rainforest is a huge system filled with many smaller systems like this, each packed with interconnected species. Every link provides stability to the next, strengthening biodiversity's weave. That weave is further reinforced by the genetic diversity within individual species, which allows them to cope with changes. Species that lack genetic diversity due to isolation or low population numbers are much more vulnerable to fluctuations caused by climate change, disease, or habitat fragmentation. Whenever a species disappears because of its weakened gene pool, a knot is untied and parts of the net disintegrate. So, what if we were to remove one species from the rainforest? Would the system fall apart? Probably not. The volume of species, their genetic diversity, and the complexity of the ecosystems form such rich biodiversity in this forest that one species gap in the weave won't cause it to unravel. The forest can stay resilient and recover from change. But that's not true in every case. In some environments, taking away just one important component can undermine the entire system. Take coral reefs, for instance. Many organisms in a reef are dependent on the coral. It provides key microhabitats, shelter, and breeding grounds for thousands of species of fish, crustaceans, and mollusks. Corals also form interdependent relationships with fungi and bacteria. The coral itself is a loom that allows the tangled net of biodiversity to be woven. That makes coral a keystone organism, one that many others depend on for their survival. So what happens when destructive fishing practices, pollution, and ocean acidification weaken coral or even kill it altogether? Exactly what you might think. The loss of this keystone species leaves its dependents at a loss too, threatening the entire fabric of the reef. Ecosystem, species, and genetic diversity together form the complex tangled weave of biodiversity that is vital for the survival of organisms on Earth. We humans are woven into this biodiversity too. When just a few strands are lost, our own well-being is threatened. Cut too many links, and we risk unraveling it all. What the future brings is unpredictable, but biodiversity can give us an insurance policy, Earth's own safety net, to safeguard our survival.
Okay, and again, that TED Ed video served just as a reminder of why it's important for us to appreciate and understand the complexity of biodiversity. And certainly species diversity is what we're kind of focusing on in our studies in 102. But the species diversity is so dependent upon genetic diversity, habitat diversity, and also that functional diversity that wasn't included in this video, so that recycling of nutrients through the ecosystem. So again, just kind of to back up and give you a reminder of why these studies are important, if you needed it. Okay, so we talked about this in part 1, so just going to kind of sum up a little bit of growth and development of animal species. So again, we're looking for those things, those ties that bind us all together to that last unknown common ancestor. So the similarities, the common thread that all animals share, and then where things are different along those lines, and why are they different, and different hypotheses that we can generate based on those molecular and morphological similarities and differences. So again, just as a reminder, if you're following along with your guided notes, hopefully this will be helpful. So a single cell generally, and these are things that are consistent throughout all animal groups, a single cell generally contains all of the genes necessary to construct the entire body, which is phenomenal. Those homeoboxes are those homogenes that we looked at earlier in part 1. There is differentiation or specialization in cells of developing embryos that occur at specific that occur with specific genes when they become activated, and those proteins are made. As the development proceeds, a single activation or certain genes produce other signals and cause this kind of cascading effect for that development of that organism in those different segments, and we see a result of those different body plans. Cells developing into embryos continue to differentiate and become specialized to perform different functions. As embryos continue to develop over time, most animals become juvies that later, that's not all of them, but some look like miniature adults, some do not. Some animals, or most animals, continue to grow into the development of those adult stages and eventually reach mature sexual stages, and the whole mating and cycle continues. Okay, this just serves as kind of a recap of the fate of the blastophore. So we're looking at two major groups of triploblastic animals, and they are differentiated into the protozoans and the deuterostomes based on the fate of the blastophore. And again, the fate of the blastophore, which happens first, is it a mouth that develops or the anus that develops? We went through the different types of development with the cleavage, differences between cleavage, the regulatory embryological development, what those embryo cells first go on to become, whether they are indeterminate or determinate, and then certainly how the coelom is formed. So here we are with our slide here. This is just hopefully useful to you in terms of organizing our different phyla that we're about to explore later on. Again, just as a reminder, mollusks and annelids and arthropods are the protozoans, and the echinoderms and the vertebrates are deuterostomes. Again, all bilateral ancestors, triploblastic organisms, but we start to see some differentiation here in terms of those early embryological stages. worth noting here, so and I know we talked just very quickly about this in part 1, but views of animal phylogeny continue to be shaped again by new technologies, new information that's acquired. And again, just as a reminder, that's why these are referred to as hypotheses. Evolution, again, is a process by which organisms will change over time. Animals form distinct branches of evolutionary tree of life. Just some reminders from the previous chapters that we've studied. The Precambrian seas started, or the origins of life, are over 600 million years ago. The hypothesis that animals probably evolved from heterotrophic protists that became colonial, and then those cells eventually became specialized but dependent upon one another for what they contributed. And again, these are things that we've already visited, how scientists hypothesize that animals first evolved in water and then later moved on to land. And then recent molecular genetics certainly support that the ancient sponge or ancient ancestors and certainly served to be the basal organism in that tree of life. Evolutionary, I shouldn't say in the tree of life, but in the evolutionary tree of animals, they serve as that basal group or outgroup. Evolutionary relationships, again, they're worked out largely based on the study of animal anatomy that we've looked at, those body plans, those differences and similarities in the DNA, and also that morphological data that includes fossils as well, so organisms that are no longer here too. Again, this just serves as kind of an illustration of, I like the way that this is organized. So we have our ancestral protists here, those that group there, and then no tissue. So we have our sponge species over here. We start to see the development of true tissue. So a two-tissue system gives us radial symmetry, which gives us things like our jellyfish or other cnidarians. Bilateral symmetry, we start to see, we differentiate here. So we have acoelomates to pseudocoelomates to coelomates. And then our coelomates start to differentiate into protozomes, which go into different types of segmentation here, and then deuterosomes, which have a different type of development as well, and different segmentation as well. So again, just kind of a nice overview of how animal groups are separated from one another, or distinct differences in those characteristics that we've already studied. Okay. Again, just kind of a reminder, most animal phyla with members alive today were established, it's estimated about 500 million years ago. There are about 3 dozen animal phyla that are recognized. And again, phylogenies are now based on the combination of whole genetic or whole genome analysis, which include morphological traits, rRNA genes, those Hox genes, protein coding genes, and mitochondrial genes as well. But having said that, there are 5 major important points to know about the relationships of extant, which, remember, extant means living animals, and these are really important to point out as we reflect upon the current phylogeny hypotheses. So let's take a quick look here, okay? So again, these are our 5 major points that we need to keep in mind as we're going through our evolutionary histories and relatedness. All animals share that common ancestor, so that ancestral protist. Sponges are a sister group to all other animals. They serve to be that outgroup or basal group. Eumetazoans, these are referred to as true animals. So again, that word eu meaning true, metazoa meaning meta meaning or zoa meaning animal. So this is a clade of animals with tissues. Okay. Most animals are bilateral. There are 3 major clades of bilateral animals, all of which are invertebrates, animals that lack backbones, except for the chordates, which are vertebrate species, which include animals like us. Okay. So we're going to dig, again, just a little bit deeper here and look at these 3 distinct clades within bilateral organisms, and this is where we focus a lot of our future studies in. So what makes them different from one another? So deuterosomes, this clade again includes both vertebrates and invertebrate species, which makes it a little different from others. The ecdysozoans and the leucotrophozoans are composed entirely of invertebrate species. The members of the ecdyzoa secrete internal skeletons. They're easy to remember because these are animals that have to shed in order to grow. And that's referred to ecdydice when they do this. So these are things you're familiar with. So these are the exoskeletons of insects. This would be the molting of things like blue crabs. They have to molt in order to grow. So grouping may include morphological related structures that are only seen when the organism is in its larval stage, and then others are within that match and align with animals throughout their entire life cycle. So leucotrophozoans are kind of an odd mix here. So a characteristic would be a leucotrophozoa is, again, they're bilateral invertebrates. Some of them have the reason why they are grouped together is during some stage of their life form, they have a feeding structure called a lophophore. They may not keep it throughout their entire life cycle, but generally, and usually it's in that juvie stage, they have a lophophore. And again, it's for acquiring nutrients in the environment. Others go through distinct developmental stages called the trochophore larvae. So they'll have this distinct stage here. Okay, again, this is just a summary slide here, just to show you again how they are, how our animal groups are differentiated from one another. So metazoa meaning animals, eumetazoa, and then our bilateral organisms here. So these just kind of cascade into one another. So eumetazoas would be the ones that have the true tissues here. So whether it's a 2-tissue system or a 3-tissue system. Then we see our bilateral, which are most animals. You can see here, this is how this is differentiated, and includes those tripoblastic organisms, those 3 germ layers, which includes deuterosomes, leucotrophozoans, and ecdyzoas. And so, again, we'll get into each one of these in more detail as we move through our upcoming chapters. This was just to serve as an overview. The rest of these slides I'll just leave it for you to go through as you wish if you want to. These are just different branches of biology. I'm just going to kind of zoom through to show you what's available here. Different fields, different terms you might hear like limnology, what is that? Parasitology, paleontology. Again, this is just an overview of different branches that you might be interested in pursuing. And then these kind of weird terms, like we heard mycologist for people who study fungus. So these guys have actual and gals have different types of terms that might sound kind of strange. Entomologists, you probably heard of that before, and apologists just studies honeybees, for example. Lepidopterans or lepidoptery is the study of butterflies and moths. Different branches of vertebrate species, like ichthyology is a course we actually offer here, so you might be interested in that. It's a study of fish. Herpetology is another course that we offer, so it's a study of amphibians and reptiles. Okay, you can even study things like cryptozoology, so these are kind of those kind of hidden organisms. And of course, zoologists, veterinarians, all kinds of things. So again, this just serves to show you those different fields. All right, thank you so much for joining.